Hi, I'm doing cell cycle as well as mitosis, and my name is Joseph O'Heather. Alright, so looking into the cell cycle, we have it broke down into two main parts. The first main part is going to be interphase, but that second crucial part is going to be mitosis. So we're going to be spending roughly 90% of the time of cell cycle in interphase, and the remainder 10% in mitosis. Okay, so looking in interphase, as you can kind of tell in the image, there's a lot going on. This is because interphase is broken down into three parts. We have the G1, which consists of a lot of the growth. We have the S phase, which is where we see a lot of that centrosine replication. As you can tell in the image, it's breaking apart and kind of like spreading off into the two ends. And then we also have G2, which consists of the cell division prep. Now, when you look inside of the nucleus, you notice that there's a lot going on in there. It's because a lot of that chromatin and everything's kind of just getting developed and ready for that next phase. Okay, so going into the mitosis phase, we're going to also see that there's five subgroups. The first phase is going to be prophase, leading into prometaphase, then metaphase, then anaphase, and lastly finishing up is going to be telophase. So for the first phase, as you see right here, prophase, we're dealing with the start where you notice there's still fragments of a nuclear envelope right here enclosing everything inside of the nucleus. And if you look, you can tell how there are sister chromatins where there's almost the duplicates from the interface that we just got set up from. And this is why there's now two pink and two blues. So what you really want to focus though on this phase is now that that centrosine that was developing and duplicating in the S phase has now actually branched off completely and is completely on both ends. And what you also want to look down and notice is that we see that there's a mitotic spindle being developed. This right here is going to be used in the next phases that we're going to be looking at. So the next phase we're going to be talking about is prometaphase. Like how I mentioned about that spindle, if you notice, it's now completely overbranched all over the sister chromatins, and it's actually completely developed. Now, if you also notice, we no longer have a nuclear envelope. That's just because it's kind of diminished and it's kind of like not needed right now, as you'll see why. At this point, we've also developed kinotromes. This is basically where the sister chromatins in the very center part where they're attached have now connected to the spindles. And if you notice, we actually see that each sister chromatin is developed on a different spindle line, and they're all attached all over. Okay, now going into metaphase, you're going to notice that we've now developed a mid plate, also known as the metaphase plate. My bad, it's also called the mid plane. This right here is where we notice all of our sister chromatin developing and lining up to, while they're still attached, of course, to our spindles. Now, if you notice, it's going from a blue to a pink to a blue to a pink, so it's almost alternating again. Okay, so at this point, now we have it to where they're all lined up, they're all attached, there's no nuclear envelope, and we have been now introduced this metaphase plate. Okay, now we're reaching the very end of the phases. We're at anaphase right now, and if you notice, prior to before, there was that midplane. It is now gone, and if you notice, all of the sister chromatins are being pulled in half to where we have half of them over here with pink, blue, but then we also have the identical copy over here on the other side, pink, blue, pink, blue. This is now giving us kind of the same information on both sides, and they're both being pulled apart. Uh, if you look though into the next phase, our final phase, telophase, you start to notice how we now have that nuclear envelope developing, as well as the cleavage for it. This is because now the DNA itself has been duplicated. We have identical information on the left as well as we do on the right. And leading into Psychonesis, we now have our daughter cell. And when you look at this image, you can actually see that they're literally identical. They're the same with each other. That itself is the process of cell cycle, resulting with us first having one singular cell to us finishing off with two identical cells. And that is it for my presentation on cell cycle and mitosis.